am going to explain about some identification tests for biological samples. But what we have to do is we have to identify some materials like carbohydrates, protein, oils, and all. So today we are doing chemical test for acacia. Acacia is a what compound it is? Carbohydrates. So we have some experiments for identifying specifically for carbohydrates and some tests for acacia also is there. The aim of this experiment is to perform the chemical test for acacia, and in experiment we have to do 0.5 gram of drug is mixed with water and 0.5 gram of drug is mixed with 2 ml of ethanol. What will happen if we are mixing with water? If carbohydrate is mixing with water means what will happen? It will be soluble or insoluble? It will be soluble. Then maybe it will be acacia. So it is confirmed as a carbohydrate. And if we are mixing with ethanol, what it will be? It will be insoluble. Then next we have to do Morris test. Morris test is a specific test for identifying the presence of carbohydrates. For Morris test, we have to take 5 ml of sample solution, add 1 ml of Morris reagent and add concentrated H2SO4 along the sides of the test tube. What will happen if we are adding along the sides of the test tube? Ring will be formed in the junction of these two liquids. What will happen? A reddish brown color ring will be formed. So it will be Morris test. What is it is confirmed? Morris test is confirmed. It will be a carbohydrate. And then we have to do test for acacia. To 10% aqueous solution of drug, add 3 ml of lead acetate solution. Then it will be forming a white precipitate. That white precipitate is formed means that is confirming the presence of acacia. So for confirming the presence of acacia, we have to add 10% of aqueous solution, then add 3 ml of lead acetate solution. This test will confirm the presence of acacia. Then next test is for identifying the presence of reducing sugar. What test we will do for reducing sugar? Spelling test. For aqueous solution of drug, we are boiled with hydrochloric acid. Then it is a required volume of sodium hydroxide solution is mixed. Then equal volume of pennies A and B solution will be adding. Then what will precipitate? Brick red precipitate will be forming. This brick red precipitate is indicating the presence of reducing sugar. So Helling test is using for identifying reducing sugar. So if acacia is reducing sugar, it will show a brick red precipitate, and we can confirm that test as our presence of reducing sugar. Then next we have to do the presence of acacia. We can do with 10% aqueous solution treated with 5 gram of borax, and we have to dilute well 10% of aqueous solution treated with iodine solution. What will happen if a carbohydrate is mixed with iodine solution? A blue color will be formed. So that blue color will be presence of blue color will be indicating the presence of carbohydrate or acacia. The presence of carbohydrate will be confirmed using iodine test. Then next we have to do aqueous solution of drug, add few drops of H2O2 and add 0.5 ml of 1% alcoholic benzene. Shake well, a distinct blue color will be formed. Then that also will be confirming the presence of acacia. So we have done around 6 tests. Then 6 tests will be indicating the presence of carbohydrate, reducing sugar, or we can confirm acacia. So what test we will do for reducing sugar? Spelling test. So spelling test part we are getting observation brick red precipitate. So brick red precipitate is giving it that will be confirming the presence of reducing sugar. Then like this we have to do experiment for castor oil also. For castor oil, we know castor oil is a liquid. So for liquids we have specific test like about 5 ml of solution petroleum either is mixed when mixed with 10 ml of castor oil at 5.5 degrees Celsius. What will happen if presence of castor oil? A clear solution is formed. So that will be confirming the presence of castor oil. And to 5 ml of oil add equal volume of alcohol at 0 degrees Celsius. The liquid and clear the what will happen? That will be clear distinct solution will be formed. That will be identify the presence of castor oil. And about 15 ml of light petroleum either when mixed with 10 ml of castor oil at 15.5 degrees Celsius. Turbid mixture is formed. That also confirms the presence of castor oil. Next, we have to go for chemical test for agar. Agar also, what type of compound is agar? Carbohydrate. It's a primary metabolic carbohydrate. So, like acacia we have done test now, same like that test also we have to do for agar. So for agar solution with cold water, aqueous solution will be boiled water and it will be cooled well. What will happen if we are mixing some carbohydrate with water and we are boiling that solution and cool it? That what will happen for that? Swelling will be occurs and gel will be formed. That will be confirmed the presence of carbohydrate or presence of agar. So that is a distinct test for agar. That swelling test is distinct for agar. Then we have to do reducing sugar test. As we mentioned in acacia, what test we have to do for reducing sugar? Swelling test. So here same we have to do that mixture of equal volume of dilute hydrochloric acid. A solution of agar is heated in water, but we have to add both types of swelling solutions. That will be observation will be red precipitate. That red precipitate will be indicating the presence of reducing sugar. Then for agar we have a specific test like test for sulfate. Aqueous solution of agar add equal volume of dilute hydrochloric acid, then treated on water bath for 30 minutes, then add barium chloride solution. What will be the observation? A white precipitate is formed. That white precipitate will be indicating the presence of acacia. So for acacia we have this specific test for sulfate. For that we have to do aqueous solution of agar, equal volume of hydrochloric acid, that we have to keep it in a water bath and we have to add hydrochloric acid and barium chloride solution, that will be forming a white precipitate. Then for agar solution add a ruthenium red solution and red color formed due to the presence of acacia. So ruthenium red is adding to the aqueous solution, then it will be forming a red color, means that is indicating the presence of acacia. Then aqueous solution of agar treated with sodium hydroxide and iodine solution. What will happen? Iodine solution, it is like deep crimson to brown color is formed. That also a specific test for agar. Then that is indicating the presence of agar. Then aqueous solution of agar is treated with tannic acid. No precipitate is formed. That is the absence of gelatin in agar. So that is indicating the absence of gelatin. That is confirming the presence of agar. Do you understand all these tests? So we have to do 3 experiments today. Identification of acacia, identification of castor oil and identification of agar. And in this acacia we have to do specifically reducing sugar test, that is swelling test, and also we have to do test for carbohydrate, solubility test we have to do, and also we have to go for reducing sugar test. And for castor oil we have to see solubility of this castor oil, and also usually we have to do with petroleum either, that how this layers is formed, that will be confirmed the presence of castor oil. And next we have to go for test for agar. For test for agar, specifically we have the test for carbohydrate, and first we have to do for gelling property. Gelling property of this test for agar. Then what is the last test we have mentioned? For the absence of gelatin. For absence of gelatin we have to treat it with what we have to treat with tannic acid. We have to treat that will be indicating the absence of gelatin, that is confirming the presence of agar. So today we are going for 3 experiments. total 3 experiments we have to complete. So, and after that we have to go for chemical test for Prakagan, chemical test for starch and all. So today we are doing these 3 experiments.